Hello everyone. Today's video is based on the tissues, chapter 6, class 9. Before starting this video, please like and subscribe to my channel LinkedIn Farmer. Let's start. Introduction. Tissues are fundamental building blocks of multicellular organisms, serving as the structural and functional units that contribute to the organization and functioning of various biological systems. Composed of specialized cells with similar structures and functions, tissues work collectively to carry out essential tasks, ranging from providing support and protection to enabling efficient communication and coordination within the organism. The study of tissues, known as histology, is crucial for understanding the intricate mechanisms that underlie the complexity of living organisms. Are plants and animals made of the same types of tissue? Plants and animals are both multicellular organisms, but they exhibit distinct differences in terms of their tissue types and organization. While both plants and animals are multicellular organisms, they differ in their organization and composition of tissues. While there are similarities between the two, there are also distinct differences in the types of tissues they possess. Comparison. While both plants and animals have tissues that serve similar functions, the specific types of tissues and their structures vary due to the distinct needs of each organism. For instance, plants require specialized tissues for photosynthesis, water transport, and support, which are not found in animals. Similarly, animal tissues like muscle and nervous tissue are unique to their physiological needs for movement and rapid communication. In summary, while plants and animals share some general functions and tissue types, their specific tissue compositions differ significantly to suit their respective biological requirements and lifestyles. Plant tissues. Plant tissues are groups of specialized cells with similar structures and functions that work together to perform specific tasks within a plant. These tissues are the building blocks of plant organs and systems, enabling plants to carry out essential functions for growth, development, and survival. 1. Dermal tissue. Dermal tissue is the outermost layer of a plant and serves as a protective barrier against environmental stresses. It includes epidermis, the outermost layer of cells that covers plant surfaces. It provides protection and helps reduce water loss through its waxy cuticle. Periderm. Found in older woody plants, periderm replaces the epidermis in areas where secondary growth occurs. 2. Ground tissue. Ground tissue makes up the majority of a plant and is responsible for various functions, including photosynthesis, storage, and support. It includes parenchyma. These cells are involved in photosynthesis, storage, and secretion. They have thin cell walls and can differentiate into other cell types. Parenchyma. Cells with unevenly thickened cell walls that provide flexible support to growing plant parts. Sclerenchyma. Cells with heavily thickened, rigid cell walls that offer structural support. They can be either fibers or sclerates. 3. Vascular tissue. Vascular tissue is responsible for transporting fluids and nutrients throughout the plant. It includes xylem, composed of vessel elements and tracheids. Xylem transports water and minerals from roots to the rest of the plant. It also provides structural support. Phloem. Made up of sieve tube elements and companion cells, phloem transports sugars, hormones, and other nutrients from the leaves to other parts of the plant. 4. Meristematic tissue. Meristematic tissue is a type of plant tissue that consists of actively dividing cells and serves as the primary location of growth and development in plants. This tissue is responsible for generating new cells, which then differentiate into various specialized cell types and contribute to the overall growth and formation of plant organs. Key characteristics of meristematic tissue include cell division. Meristematic cells are continually undergoing cell division, mainly through mitosis. This division leads to the production of new cells that contribute to the increase in plant size. Undifferentiated cells. Meristematic cells are undifferentiated, meaning they haven't yet acquired specific structures or functions associated with mature cell types. As they divide, some of these new cells will differentiate into specialized cell types, such as parenchyma, parenchyma, sclerenchyma, xylem, and phloem cells. Location. Meristematic tissue is typically found in areas of a plant where growth occurs. The two main types of meristematic tissue are apical meristems and lateral meristems. Apical meristems. These are found at the tips of roots and stems. They are responsible for primary growth, which leads to an increase in the length of the plant. The apical meristem at the root tip is called the root apical meristem, while the one at the stem tip is called the shoot apical meristem. Lateral meristems. These meristems are responsible for secondary growth, which leads to an increase in the girth or thickness of stems and roots. Lateral meristems include the vascular cambium and cork cambium. Meristematic tissue is crucial for the development and maintenance of plant structures. As meristematic cells divide and differentiate, they give rise to all the various cell types necessary for plant growth, such as those found in leaves, flowers, stems, and roots. Without meristematic tissue, plants would not be able to adapt to changing environments, repair damage, or achieve their characteristic shapes and sizes. Activity. Growth of roots in onion bulbs. Initial swelling. When an onion bulb is planted in soil or water, it undergoes an initial swelling phase. This is due to the absorption of water by the outer layers of the bulb. Meristematic activation. The root apical meristem in the onion bulb becomes activated. Meristematic cells in this region start dividing rapidly, leading to the formation of new cells. Cell elongation. As meristematic cells divide, some of the newly formed cells undergo elongation. This elongation is responsible for the growth in length of the developing roots. Root hair formation. Specialized structures called root hairs begin to develop on the surface of the elongating root cells. Root hairs significantly increase the surface area of the root, enhancing its ability to absorb water and nutrients from the soil. Cell differentiation. As the cells continue to divide and elongate, they start differentiating into various specialized cell types, including epidermal cells, cortex cells, and vascular tissues, xylem and phloem. Gravity response. The direction of root growth is influenced by gravity. The root tip, known as the root cap, senses gravity and guides the root's growth downward, a process known as positive geotropism. Nutrient and water absorption. The root system continues to grow and develop more root hairs. These root hairs play a vital role in absorbing water and nutrients from the soil, which are essential for the plant's overall growth and development. Anchorage and support. As the root system expands, it provides anchorage and support to the growing onion plant. The roots penetrate the soil and hold the plant in place, preventing it from being easily uprooted by wind or other external forces. Continued growth. Root growth in onion bulbs is ongoing as long as the plant has access to the necessary resources, including water, nutrients, and suitable environmental conditions. Integration with shoot growth. The growth of roots and shoots, above ground parts, is interconnected. The root system provides water and nutrients to the rest of the plant, supporting its overall health and development. In summary, the growth of roots in onion bulbs involves a sequence of processes, including cell division, elongation, differentiation, and specialized adaptations like root hair formation. This growth is crucial for nutrient absorption, anchorage, and overall plant well-being. 5. Permanent tissue. Permanent tissue is a type of plant tissue that consists of cells that have completed their growth and differentiation and have assumed their final form and function. Unlike meristematic tissue, which consists of actively dividing cells, permanent tissue is composed of cells that are no longer actively dividing but have become specialized for specific roles within the plant. Permanent tissues can be further classified into two main types: simple permanent tissues and complex permanent tissues. E. Simple permanent tissues. Parenchyma, collenchyma, sclerenchyma. B. Complex permanent tissues. Xylem, phloem. Permanent tissues play a crucial role in the overall function and structure of plants. They provide support, carry out various metabolic processes, and facilitate the transportation of water, nutrients, and sugars. The distinction between simple and complex permanent tissues reflects the diversity of cell types and functions necessary for plants to adapt to their environments and fulfill their life processes. Activity: observing permanent tissues in plant slides. In this activity, you can observe permanent tissues in plant slides under a microscope. This hands-on exploration will help you better understand the diversity of cell types and their functions within plant tissues. Materials needed: microscope, plant tissue slides, prepared microscope slides, cover slips, microscope slide holder or stage. Instructions. Preparation: obtain plant tissue slides from a biology lab or educational supply store. These slides may contain cross-sections of plant organs, such as stems, roots, or leaves, that showcase different types of permanent tissues. Microscope setup: set up your microscope on a stable surface. Adjust the light intensity and focus of the microscope to ensure clear visibility. Slide placement: place a plant tissue slide on the microscope stage. Secure it in place using the slide holder or stage clips. Observation: start with low magnification, examples 10x, and observe the entire slide to get an overview of the tissue arrangement. Then, switch to higher magnifications, examples 40x or 100x, to focus on specific cell types and structures. Identify tissues: look for areas on the slide that contain different types of permanent tissues. Focus on areas that display variations in cell shape, size, arrangement, and staining. Simple permanent tissues: identify and observe the different types of simple permanent tissues present on the slide, such as parenchyma, collenchyma, and sclerenchyma. Pay attention to their cell wall thickness, shape, and arrangement. Complex permanent tissues: locate sections of xylem and phloem tissues within the slide. Observe the different cell types within these complex tissues, such as vessel elements, tracheids, sieve tube elements, and companion cells. Take note of their roles in water and nutrient transport. Recording observations: use a notebook to sketch and label what you observe. Note the differences in cell types, shapes, and functions in the different tissues. Cover slips: if desired, you can carefully place a cover slip over the tissue slide to prevent it from drying out and to improve clarity. Gently press down on the cover slip to remove any air bubbles. Discussion: after your observations, reflect on the significance of the observed tissues in plant growth, development, and overall function. Consider how the various permanent tissues work together to support the plant's life processes. This activity will allow you to connect theoretical knowledge about permanent tissues with real-life observations. It's a great way to enhance your understanding of plant anatomy and the roles different tissues play in plant biology. Simple permanent tissues. Simple permanent tissues are groups of plant cells that have completed their growth and are specialized for specific functions. Unlike meristematic tissues, which are actively dividing, simple permanent tissues consist of mature cells that have ceased to divide and have taken on distinct roles within the plant. There are three main types of simple permanent tissues: parenchyma, collenchyma, and sclerenchyma. 1. Parenchyma tissue. Characteristics: parenchyma cells are characterized by thin cell walls and living protoplasts, cell contents. These cells are versatile and have the ability to differentiate into other cell types if needed. Function: parenchyma tissue serves various functions, including photosynthesis, storage of nutrients, examples, starch, and secretion. They also contribute to healing and tissue regeneration. Location: parenchyma cells are found in various plant organs, such as leaves, mesophyll, stems, cortex, roots, cortex, and fruits. In leaves, they are responsible for photosynthesis. 2. Collenchyma tissue. Characteristics: collenchyma cells have unevenly thickened primary cell walls, primarily in the corners where cells are in contact with each other. This gives them flexibility and support. Function: collenchyma tissue provides mechanical support to growing plant parts. It helps in withstanding bending and stretching forces that young plant organs often experience. Location: collenchyma cells are usually found beneath the epidermis in stems and leaves of young plants. They are particularly abundant in regions undergoing active growth. 3. Sclerenchyma tissue. Characteristics: sclerenchyma cells have heavily thickened, lignified secondary cell walls that provide great strength and rigidity. They are often dead at maturity. Function: Sclerenchyma tissue provides strong structural support to plant organs. They also help in protection against herbivores and environmental stresses. Location: Sclerenchyma cells are commonly found in regions that require additional mechanical strength, such as the vascular bundles, seed coats, and stems of mature plants. Summary: simple permanent tissues play critical roles in the overall structure and functioning of plants. Parenchyma cells support photosynthesis, storage, and healing. Parenchyma cells offer flexible support during growth, and sclerenchyma cells provide rigid structural support and protection. The diversity of functions and cell types within simple permanent tissues showcases the adaptability of plants to their environment and the coordination needed for their survival and growth. Activity: investigating simple permanent tissues in plant parts. In this activity, you will explore different plant parts to observe and identify the various types of simple permanent tissues: parenchyma, parenchyma, and sclerenchyma. By using readily available plant samples, you can enhance your understanding of how these tissues function within a plant's structure. Materials needed: fresh plant samples, leaves, stems, and roots, hand lens or magnifying glass, scalpel or sharp knife, cutting board, microscope slides and cover slips, microscope optional. Instructions: sample collection: collect samples of leaves, stems, and roots from various plants. Choose young and mature plant parts to observe differences in tissue structure. Leaf observation: A. Examine a leaf under natural light or using a hand lens. Observe the transparent areas of the leaf, which are typically parenchyma cells involved in photosynthesis. B. If available, gently peel off a thin layer of leaf tissue using a scalpel or knife. Place the tissue on a microscope slide, add a drop of water, and cover with a cover slip. C. Observe the prepared slide under a microscope, if available, to see the arrangement of parenchyma cells and any chloroplasts they may contain. Stem observation: A. Examine a cross-section of a young stem using a hand lens or magnifying glass. Look for regions of green color, indicating parenchyma cells, and areas with elongated cells, indicating parenchyma cells. B. Carefully cut a thin cross-section of a stem using a scalpel or knife. Place the section on a microscope slide, add a drop of water, and cover with a cover slip. C. Observe the prepared slide under a microscope, if available, to see the arrangement and characteristics of parenchyma cells. Root observation: A. Examine the outer layer of a root, which may appear darker and tougher due to sclerenchyma cells. B. Cut a small section of the root and place it on a microscope slide. Add a drop of water and cover with a cover slip. C. Observe the prepared slide under a microscope, if available, to examine the structure of sclerenchyma cells. Comparison and discussion: compare your observations of parenchyma, parenchyma, and sclerenchyma tissues in different plant parts. Discuss their functions and roles within the plant's overall structure and growth. Documentation: record your observations in a notebook, including sketches, labels, and descriptions of the tissues you observed. Take note of any differences you observed between young and mature plant parts. By directly observing and identifying these simple permanent tissues in various plant parts, you'll gain a deeper appreciation for their diverse roles in supporting the plant's physiological processes and structural integrity. This hands-on activity will enhance your understanding of plant anatomy and tissue diversity. B. Complex permanent tissues. Complex permanent tissues are groups of different types of cells that work together to perform specific functions within a plant. These tissues are called complex because they are composed of multiple cell types that collaborate to fulfill vital roles in plant growth, nutrient transport, and support. The two main types of complex permanent tissues are xylem and phloem. 1. Xylem tissue. Characteristics: xylem tissue is responsible for the transport of water, minerals, and dissolved nutrients from the roots to other parts of the plant. It is made up of several cell types, including vessel elements, tracheids, fibers, and parenchyma cells. Vessel elements: these are tube-like cells with perforated end walls called perforations. Vessel elements allow for efficient water transport. Tracheids. Tracheids are long, tapered cells with smaller pits that enable water movement. They are common in gymnosperms and some angiosperms. Fibers. Fibers provide mechanical support to the xylem tissue. They have thick, lignified walls. Parenchyma cells. Parenchyma cells store nutrients and contribute to lateral transport within the xylem tissue. Function. Xylem tissue conducts water and minerals from the roots to the aerial parts of the plant. It also provides structural support. 2. Phloem tissue. Characteristics. Phloem tissue is responsible for transporting sugars, mainly sucrose, hormones, and other organic molecules throughout the plant. It consists of sieve tube elements, companion cells, fibers, and parenchyma cells. Sieve tube elements. These are tube-like cells with perforated sieve plates at their ends. Sieve tube elements are responsible for sugar transport. Companion cells. Companion cells are connected to sieve tube elements and provide metabolic support for them. They help load and unload sugars. Fibers. Similar to xylem, fibers in phloem offer mechanical support to the tissue. Parenchyma cells. Parenchyma cells in phloem store and transport nutrients and contribute to tissue flexibility. Function. Phloem tissue transports the products of photosynthesis, sugars, from source regions, examples, leaves, to sink regions, examples, roots, developing fruits. It also plays a role in hormonal signaling and nutrient distribution. Summary. Complex permanent tissues, such as xylem and phloem, are crucial for plant survival and growth. Xylem facilitates the movement of water and minerals, while phloem transports nutrients and signals throughout the plant. The collaboration of different cell types within these tissues ensures the efficient distribution of resources and information, enabling plants to adapt to changing conditions and achieve optimal growth and development. Activity. Studying xylem and phloem tissues in plant stems. This activity will allow you to investigate complex permanent tissues, specifically xylem and phloem, in plant stems. By preparing and examining stem cross-sections, you'll gain a deeper understanding of how these tissues function in transporting water, nutrients, and sugars within the plant. Materials needed. Fresh plant stems, examples, celery, sunflower, or other available plants, scalpel or sharp knife, cutting board, hand lens or magnifying glass, microscope slides and cover slips, microscope optional. Instructions. Sample collection. Obtain fresh plant stems from different plants. Choose stems with different sizes and ages for a comprehensive understanding of tissue variation. Stem cross-section preparation. A. Place a stem on a cutting board. B. Use a scalpel or sharp knife to make a clean, thin cross-section near the base of the stem. Be careful not to crush or damage the tissues. C. Transfer the cross-section to a clean microscope slide. If desired, add a drop of water to the section to prevent it from drying out. Observation without microscope. Examine the stem cross-section using a hand lens or magnifying glass. Observe the arrangement of different tissues, including the outer epidermis, cortex, vascular bundles, and pith. Xylem observation. A. Look for small, distinct circular or oval regions within the vascular bundles. These are the xylem vessels and tracheids. B. Note the presence of darker areas within the xylem, indicating the lignified cell walls. Phloem observation. A. Identify the phloem regions next to the xylem. Phloem cells are typically more elongated than xylem cells. B. Look for cells with perforated end walls, sieve plates, within the phloem. These are the sieve tube elements. Microscopic observation, optional. If you have access to a microscope, carefully place a cover slip over the stem cross-section on the microscope slide. Observe the slide under low and high magnification to see the cellular details of xylem and phloem tissues. Documentation. Record your observations in a notebook. Draw labeled sketches of the stem cross-section, highlighting the xylem and phloem tissues. Include notes on cell types and structures. Discussion. Reflect on your observations and discuss the role of xylem and phloem tissues in the overall functioning of the plant. Consider their functions in water and nutrient transport and the significance of their arrangement within the stem. By engaging in this activity, you'll develop a hands-on appreciation for the complex permanent tissues that play a pivotal role in the survival and growth of plants. Understanding the functions and structures of xylem and phloem will deepen your knowledge of plant anatomy and physiology. Animal tissues. Animal tissues are specialized groups of cells with similar structures and functions that work together to perform specific tasks within the bodies of animals. These tissues are the fundamental building blocks of animal organs and systems, enabling various physiological processes necessary for the survival and functioning of the organism. Animal tissues are categorized into 4 primary types based on their functions and characteristics: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Each type serves a unique purpose and contributes to the overall complexity of animal body organization. 1. Epithelial tissue. Epithelial tissue is one of the primary types of animal tissues, and it covers the surfaces of the body, lines body cavities, and forms the outermost layers of organs. Epithelial tissue serves various functions, including protection, absorption, secretion, excretion, and sensation. It is characterized by its tightly packed cells and the absence of blood vessels within the tissue itself. Characteristics of epithelial tissue. Cellularity. Epithelial tissue consists of closely packed cells that are bound together by specialized junctions. These cells are arranged in continuous sheets or layers. Polarity. Epithelial cells have an apical, upper, surface that faces a free space or a body cavity, and a basal, lower, surface that is anchored to a basement membrane, a thin extracellular matrix. Attachment. The basal surface of epithelial cells is attached to a basement membrane, which provides support and helps anchor the tissue to underlying connective tissue. Avascularity. Epithelial tissue lacks blood vessels within the tissue itself. It receives nutrients and oxygen through diffusion from nearby blood vessels in the underlying connective tissue. Regeneration. Epithelial tissue has a high regenerative capacity. Cells at the basal layer continuously divide to replace damaged or lost cells at the apical surface. Types of epithelial tissue. There are several types of epithelial tissue based on cell shape and arrangement. Simple epithelium consists of a single layer of cells and is mainly involved in absorption, secretion, and filtration. Types include simple squamous, flat cells, simple cuboidal, cube-shaped cells, and simple columnar, column-shaped cells. Stratified epithelium comprises multiple layers of cells and provides protection against mechanical and chemical stresses. Types include stratified squamous, keratinized or non-keratinized, stratified cuboidal, and stratified columnar. Pseudostratified epithelium appears stratified but is actually a single layer of cells with varying heights. Often ciliated and found in the respiratory tract. Transitional epithelium, found in organs like the urinary bladder, this tissue can stretch to accommodate changes in organ volume. Functions of epithelial tissue. Protection. Epithelial tissue forms a barrier that protects underlying tissues from physical injury, microbial invasion, and dehydration. Absorption. Epithelial cells can absorb nutrients, ions, and water across their surfaces, aiding in digestion and nutrient uptake. Secretion. Glandular epithelial cells produce and release substances such as enzymes, hormones, mucus, and sweat. Excretion. Certain epithelial cells eliminate waste products from the body, such as in the kidneys. Sensation. Some epithelial cells have sensory functions, detecting stimuli like touch, pressure, temperature, and taste. Summary. Epithelial tissue is essential for maintaining the integrity and functionality of animal bodies. Its diverse functions, shapes, and arrangements allow it to fulfill a wide range of roles, from protecting internal structures to facilitating absorption, secretion, and sensory perception. The organization of epithelial cells into layers and sheets contributes to the complex and specialized functions of different organs and body systems. 2. Connective tissue. Connective tissue is a diverse and abundant type of animal tissue that serves to connect, support, protect, and bind together various structures within the body. It is found throughout the body, ranging from the outermost skin to the innermost organs. Connective tissue plays a crucial role in maintaining the structural integrity of the body and providing a framework for other tissues and organs to function. Characteristics of connective tissue. Matrix. Connective tissue is characterized by an extracellular matrix, which consists of fibers, collagen, elastic, reticular, and a ground substance, a gel-like material that varies in consistency. The matrix gives connective tissue its strength, flexibility, and other functional properties. Cell types. Connective tissue contains various types of cells, including fibroblasts, responsible for producing fibers, adipocytes, fat-storing cells, macrophages, immune cells, and specialized cells like osteocytes, bone cells, and chondrocytes, cartilage cells. Vascularity. Vascularity varies among different types of connective tissue. Some types, like blood and bone, are well vascularized, while others, like cartilage, have a limited blood supply. Variability. Connective tissue is incredibly diverse, with different types varying in terms of cell types, fibers, and ground substance composition. Types of connective tissue. There are several types of connective tissue, each with its own characteristics and functions. 1. Connective tissue proper. Loose connective tissue comprises fibers and cells loosely arranged in a gel-like matrix. Includes areolar tissue, support and protection, and adipose tissue, fat storage and insulation. Dense connective tissue contains densely packed collagen fibers, providing strength and resistance to stretching. Includes dense regular, tendons and ligaments, and dense irregular, dermis of skin tissues. 2. Supportive connective tissue. Cartilage consists of chondrocytes embedded in a firm, flexible matrix. Types include hyaline cartilage, covers joint surfaces, elastic cartilage, flexibility, e.g., ear, and fibrocartilage, shock absorption, e.g., intervertebral discs. Bone, osseous tissue, contains osteocytes within a matrix of collagen fibers and mineral deposits, calcium phosphate. Provides support, protection, and mineral storage. 3. Fluid connective tissue. Blood, composed of red blood cells, white blood cells, platelets, and plasma. Functions in transport, immune response, and clotting. Functions of connective tissue. Support and structure. Connective tissue forms the structural framework for various organs and tissues, providing support and maintaining their shapes. Protection. Connective tissue cushions and protects delicate organs and structures from mechanical stress and impact. Transport. Blood, a fluid connective tissue, transports oxygen, nutrients, hormones, and waste products throughout the body. Energy storage. Adipose tissue stores energy in the form of fat for later use. Immune response. Macrophages and other immune cells in connective tissue help defend the body against pathogens and foreign particles. Healing and repair. Connective tissue plays a key role in wound healing and tissue repair by forming scar tissue. Summary. Connective tissue is a versatile and vital component of the animal body, providing structural support, protection, energy storage, and transportation. Its wide variety of types and functions ensures that the body's different structures and systems work together seamlessly. Connective tissue's unique combination of cells, fibers, and extracellular matrix contributes to its specialized roles in maintaining the body's integrity and functionality. 3. Muscle tissue. Muscle tissue is a specialized type of animal tissue that is responsible for generating force and producing movement within the body. It allows animals to perform various functions, from simple actions like blinking to complex movements like walking and running. Muscle tissue is categorized into 3 main types: skeletal muscle, smooth muscle, and cardiac muscle. Characteristics of muscle tissue. 1. Excitability. Muscle cells, called muscle fibers, are able to respond to stimuli, such as nerve signals, by generating electrical impulses. 2. Contractility. Muscle tissue has the unique ability to contract, generating force and causing movement. This is achieved through the interaction of protein filaments within muscle fibers. 3. Extensibility. Muscles can be stretched and extended without being damaged. This is important for maintaining flexibility and allowing movement in different directions. 4. Elasticity. After being stretched, muscle tissue can return to its original shape and length. This property ensures that muscles can resume their functions after contraction or extension. Types of muscle tissue. 1. Skeletal muscle. Structure. Skeletal muscle tissue is composed of long, cylindrical, multinucleated muscle fibers that are striated, striped in appearance due to the arrangement of protein filaments. Control. Skeletal muscles are under voluntary control, meaning they can be consciously controlled by the nervous system. Function. Skeletal muscles are responsible for body movement, posture maintenance, and generating heat through contractions. They are attached to bones via tendons. 2. Smooth muscle. Structure. Smooth muscle tissue consists of spindle-shaped, non-striated cells with a single nucleus. These cells are found in the walls of internal organs and blood vessels. Control. Smooth muscles are involuntary and are controlled by the autonomic nervous system. Function. Smooth muscles regulate movement within hollow organs, such as the digestive tract, blood vessels, and respiratory airways. They help propel substances through these structures. 3. Cardiac muscle. Structure. Cardiac muscle tissue is found in the heart and consists of branching, striated cells connected by intercalated discs. These discs allow for synchronized contractions. Control. Cardiac muscle is involuntary and is regulated by the autonomic nervous system and the heart's internal pacemaker. Function. Cardiac muscles contract rhythmically to pump blood throughout the circulatory system. The heart's continuous, coordinated contractions are essential for maintaining blood flow. Functions of muscle tissue. Body movement. Skeletal muscles generate forces that cause movement at joints, allowing animals to walk, run, jump, and perform other motions. Posture maintenance. Muscle contractions help maintain body posture by balancing opposing forces around joints. Heat generation. Skeletal muscles generate heat as a byproduct of energy production during contraction, contributing to body temperature regulation. Organ function. Smooth muscles control the movement of substances within various internal organs, facilitating processes like digestion and blood circulation. Heart contraction. Cardiac muscles enable the heart to beat rhythmically, pumping blood and maintaining circulation. Summary. Muscle tissue is essential for enabling movement, maintaining posture, and facilitating the functions of organs like the heart, blood vessels, and digestive tract. Its unique ability to contract, along with its different types and properties, allows animals to carry out a wide range of physical activities necessary for survival and daily life. 4. Nervous tissue. Nervous tissue is a specialized type of animal tissue that forms the communication network within the body. It is responsible for transmitting electrical signals, known as nerve impulses or action potentials, which allow animals to perceive, process, and respond to information from their environment. Nervous tissue is the foundation of the nervous system, which includes the brain, spinal cord, and nerves. Characteristics of nervous tissue. 1. Neurons. Neurons are the primary cells of nervous tissue. They are specialized to transmit nerve impulses. Neurons consist of a cell body, somer, dendrites, receiving incoming signals, and an axon, conducting outgoing signals. 2. Glial cells. Glial cells, also known as neurolia or glia, are supportive cells that surround and protect neurons. They assist in maintaining the environment around neurons and provide structural support. 3. Electrical signaling. Nervous tissue communicates using electrical signals called action potentials. These signals are generated in response to stimuli and are conducted along axons to transmit information. 4. Synapses. Neurons communicate with each other and with target cells, such as muscles or glands, at specialized junctions called synapses. Neurotransmitters are released at synapses to transmit signals from one neuron to another or to the target cell. Types of nervous tissue cells. 1. Neurons. Sensory neurons transmit sensory information, e.g., touch, temperature, pain, from sensory receptors to the central nervous system, CNS. Interneurons, association neurons, found within the CNS, these neurons process and integrate incoming signals, making decisions about responses. Motor neurons transmit signals from the CNS to muscles or glands, initiating muscle contractions or glandular secretions. 2. Glial cells, astrocytes, provide support to neurons, maintain the chemical environment around them, and contribute to the blood-brain barrier. Microglia act as immune cells within the CNS, protecting against pathogens and debris. Oligodendrocytes, CNS, and Schwann cells, PNS, wrap around axons, forming myelin sheaths that insulate and increase the speed of action potential conduction. Ependymal cells line fluid-filled cavities in the CNS and assist in the circulation of cerebrospinal fluid. Functions of nervous tissue. Sensory input. Nervous tissue receives sensory information from the environment and transmits it to the central nervous system, CNS, for processing. Integration. The CNS processes and interprets sensory information, makes decisions, and initiates responses. Motor output. Nervous tissue transmits commands from the CNS to effectors such as muscles and glands, resulting in appropriate responses. Homeostasis. Nervous tissue helps regulate the body's internal environment, ensuring proper physiological conditions are maintained. Learning and memory. Nervous tissue is involved in learning and memory processes, enabling animals to adapt to new information and experiences. Summary. Nervous tissue is a complex and essential component of the animal body, enabling communication, perception, coordination, and response. Neurons transmit electrical signals, while glial cells provide support and protection. The integration of sensory input, processing, decision-making, and motor output underlies the nervous system's intricate ability to control and regulate various physiological functions. Thank you for watching. Please like, share and subscribe.